When you're using something to build a drug, you're kind of asking the model to, in one shot, spit out Shakespeare's sonnet. It needs to be the best. You don't want to sample from the distribution of average. You actually need the short tail of the distribution. I can tell you, scientists are often not the best marketers. I think you can have like a real impact on like really meaningful stuff. At least you can work on something that's not a goddamn chatbot. So you get to do that.
Hey, Trevor.
Long time no see.
Long time no see.
All right, let's get the show on the road.
Cool. So, I mean, obviously you've done some incredible things with Mammoth. And at the time when I heard about what you're doing, it was during the whole COVID time. You guys were one of the very first people to actually build tests that really quickly detect that stuff. It was a really important problem at a pretty critical time. And it just so happened to very much benefit from the technology that you're building with CRISPR. I almost said CRISPR-Cas9, but you're doing something cooler than Cas9. Do you want to talk a little bit on that?
Yeah, sure. Yeah, no, it's interesting. The company's definitely been on a journey in terms of the different applications we've used CRISPR for. I think diagnostics is a super cool, non-obvious one, because I think when people think about CRISPR or Cas9 or whatever you want to refer to it with, you really do think about therapeutics. And that's what we focus on today, and we can get into that. But I think in the history of the company, one of the coolest things we've done is we actually found a way to use CRISPR to build diagnostic tests specifically for COVID to help out with the pandemic. And it turns out to be a really powerful way of building really low cost, easy to use, effective diagnostic tests. And that's a very non-obvious thing, I would say.
Completely agreed. I think the other thing that people don't think about with CRISPR is the importance of the Cas component and having great enzymes. You've pioneered both Cas5, Cas14, I believe, a bunch of...
Yeah, the funny thing about CRISPR, because we all refer to it as CRISPR, even in the scientific community, but CRISPR, the reason why it's spelled in all caps is it's actually an acronym. It stands for Clustered Regulatory Interface Short Palindromic Repeats. But it actually has nothing to do with the protein. That's called Cas. And it can be called Cas9 or Cas or nanocas or whatever, but actually, CRISPR is an array that you find in organisms of kind of where they store the viruses that they had to chop up with CRISPR when they were using it as an immune system. But the details of it aren't important. I think the irony is that everyone refers to this technology as CRISPR when actually that's not really the component of the scientific process that's really leveraged often when you're doing therapies or diagnostics or anything. It's really the Cas component.
Yeah.
But no one calls it Cas. I think CRISPR just sounds better. I think it's the power of marketing.
Yeah.
I don't think the technology would have gotten so far if you called it Cas.
What do you think is the most important thing to think about when you're designing the Cas component in terms of, obviously the difference between Cas9 and the platform that you've built is pretty major. What would you say is the number one thing you're proud of that you and your team have done on that front?
Well, I think, so the company was spun out of Jennifer Doudna's lab at Berkeley, and Jennifer won the Nobel Prize for inventing this CRISPR technology. And one of the big problems, though, with the original one, which is called Cas9, which is probably the most famous, and it has these big patent fights where universities are kind of fighting over who really invented it. Obviously, we have strong opinions about that. But one of the big things is that it's actually a big protein. So from our perspective, we're humans, all proteins are tiny. But if you zoom down to the level of the cell, proteins can vary very wildly in size. You have, your semi-trucks, you have your smart cars, you have everything in between. And it turns out Cas9 is more of like a semi-truck. It's very large. It's really tough for it to navigate around different places. And you can think of the proteins that we've developed at Mammoth and out of Jennifer's lab and the work my co-founders Janice and Lucas did as being kind of the smart cars where they can kind of go anywhere. And that's a huge problem because there's many different types of cells in your body, there's many different tissues. And if you really want to build genetic therapies, you have to be able to deliver to any of them, maybe all of them. And that's a big problem in this space. So that's one of the big things that we've unlocked by having these really, really tiny versions, even tinier than normal protein, is that we can actually deliver them anywhere. So
from what I've last read, it's less than half the size of Cas9.
Yeah, that's less than a third.
Less than a third?
No, they can be very small. Yeah.
Impressive.
Qualitatively different realm. And yeah.
I like the truck analogy. I know you're someone that likes to think a lot from first principles. And there was a blog of yours called Short Tales.
Wow, the deep lore here.
Pretty punny title. I like what you did there. Yeah. And it actually isn't very biotech related at all. It's mostly statistics, and a little bit of history there as well.
Yeah,
Which-
I thought I would be a history major when I went to college.
How-
My first love.
So you started out wanting to do history, and then you've done a lot of statistics and data science, and now it seems like a lot of that is somehow integrated into what you're currently doing. Even in the company name, there's a bit of a historical nod there.
Yeah, for sure. Yeah, we can get into short tales if you want. It's definitely like a fun era of my life. But yeah, in general, in high school, I probably was most interested in history, but definitely like loved the sciences generally, mostly physics and chemistry. But no, not because it was taught poorly, but just the way it was taught was biology was more stamp collecting. So it's like, what's the parts of the plant, or what are the species classifications? So I never really liked biology growing up. But then when I went to college, I just kind of on a whim signed up for this program called Integrated Sciences that they had at Princeton. And it was run by this guy named David Botstein, who was a famous biologist originally from Stanford before he came over. And the whole point of the program was to trick people that were more on the physics and chemistry side of the equation and computer science and math into becoming biologists and teaching biology in this different way of like, actually, there are interesting, like repeatable rules you can learn, and it's not just like memorization or stamp collecting. And I definitely got like nerd sniped and fell in love with this concept. And first was kind of going down the physics route because I loved physics as well. And then quickly realized that I could work 100 hours a week and I would never be the best physicist. And that was, I think, a good lesson for me. But it turns out I could be a pretty good physicist, and I could be a pretty good biologist. I could be a pretty good statistician. And working at the intersection turned out to be something unique where I could offer something. So that's where I got really involved in like interdisciplinary work. And I think that now it's like more popular, but especially back in like 2007, 2008, I think that was more of a novel concept. Now you have to stop physicists from doing too much biology, especially during the pandemic. Everyone thinks they know biology, but back then I think it was a very powerful program for kicking off this kind of era of computational biology.
I think the fact that you're living in San Francisco has a lot to say about the type of company you're building. Usually when people talk about CRISPR, this is a conversation that's had in Boston,
Sure.
sometimes in San Diego, not as frequently in San Francisco, but ideally more over time. How do you think about existing, software engineers and data scientists who may not want or may not know that much about biology already, but want to be involved in this field? Do you think that's something that's possible?
Yeah, no, I think there's huge opportunities. I think when I first got started in the field, people used to talk about computational biology like it was some different field or something. And now I think no one uses that word because all biology is computational. It's like a misnomer of like, of course, you're dealing with data, often very large data sets. And then, of course, the benefit is that you get to have an impact on human health. So the downside, if I'm being honest, is I've heard the salaries that like the foundation labs are giving for AI scientists, and I can tell you biotech companies aren't paying that, but at least you can work on something that's not a goddamn chatbot. So you get to do that. So I think that's the put and the take. So I think you can have like a real impact on like really meaningful stuff. But I think biotech does have differences in terms of the industry, in terms of like, frankly, the amount of capital that's in it versus like other applications. And I think that's really fucked up, but it's just the reality of it.
I'll push back a little bit on just the chatbot thing and say,
I mean, purposefully inflammatory.
one of the things that's cool about said chatbot and these large language models is that they're text-based, which DNA happens to be as well.
Yeah.
Do you see all of the investment going into LLMs being beneficial for computational bio?
Oh yeah, for sure. Don't get me wrong. Yeah, I think you do need to train models specifically for biology, because in some ways, the good and the bad news. The good news is biology is much simpler in a way. Whereas we have, 26, I'm not an English major, letters in the human alphabet. There's actually only 4 letters for life, A, T, C, and G. So it makes up your DNA. And there's 2 billion of these letters that make up our DNA. And, we have some differences, but, roughly the same overall. And so you have a somewhat simpler problem in a way. But what's more complex, among many things, I'll give 2. One is that in language, I think that the context is a lot smaller. Like if I give you the 5 words before a word and the 5 words after, you probably can predict that word pretty well. I think the context window is pretty small in a way. Like you don't need to look 2 chapters back to try and predict the next word in the sentence.
Yeah.
Whereas in biology, I think that's fundamentally different for a lot of reasons. One is that the DNA is actually folding in 3D space. So something that's at one end of the chromosome might, in physical space, be right next to something at the other end. So your context window is kind of unknown and like kind of determined by other factors that are maybe hidden from you without doing certain measurements. So I think context is very difficult in biology compared to language. I think in language, you can make simple assumptions that turn out to be quite good. I think in biology, that's less the case. And then two, I think there's a lot of, and it's kind of related, there's a lot of context that's not in those letters, unfortunately. So the most obvious one is epigenetics. So there's things like methylation, for example, that's on top of the DNA. But there's a lot of things as well. It's just the physical location of things in the cell. There's the RNA that then is processed. So DNA goes to RNA, goes to protein, goes to phenotype. And there's a lot of things in between all those as well. And I think that's why it's not that the problem's impossible, but I think it actually is a very much more complex problem than language. I think it's much harder, actually. And there's much less data to work with.
Interesting. So if the constraint is context, and the context, I assume, is specific to each individual person and maybe different.
You might find some general rules, but it's like, whereas in language, you can just say plus or minus 5 words is a good enough context window.
Yeah.
In biology, that's probably a very incorrect assumption, actually.
So how does this impact the ability to do in silico research versus...
I think it means the problem is much earlier than other models. I think if you want to like simulate a cell, there's 1,000 things that are just unmeasured today that you would need to even measure to begin. So the bad news is I feel that we're probably a ways off from doing that. The good news is that I think in general, these models have been shockingly powerful, at least within language. So I think hopefully that also applies to like other domains, like language of life. But the other complication is that when you're using these models, not as a chatbot, but to actually, let's say, generate a drug, which is something people always talk about, right? Whereas when you're using a chatbot, the model just needs to be good enough. It needs to converse with you and give you reasonable answers. And it could be of average intelligence, and that's a perfectly fine chatbot. When you're using something to build a drug, you're kind of asking the model to, in one shot, spit out maybe 100 drugs. Let's give it some credit. Not asking for one, maybe 10,000. But it kind of needs to write Shakespeare's sonnet. It needs to be the best. And that's a pretty different problem from normal chatbot use, where you're kind of just asking it to give good enough answers. And I think we see that a little bit in biology, where some of the results are like good enough. But when you're asking like a biology LLM to generate like a new protein or new drug, if it's as good as what's already out there, that's not helpful. You already got that drug, right? So if it spits out 100 things that are like good, you don't care. You're not going to run that through a clinical trial. You need to spit out something that's 100 times better. And it's a bit of a different way of viewing, in my opinion, the use of these LLMs. It's like, you need to just spit out the great American novel when you ask it something. It can't just give you something that's okay. And I think that's a different use.
Got it. Interesting. So, yeah, I mean, I guess it trends towards the norm, or there's sort of this like stochastic.
Yeah, you don't want to sample from the distribution of average. You actually need the short tail of the distribution.
It seems like you like short tails.
Yeah.
Interesting.
Outliers, so.
The importance of creating incentives in regulation, not just to minimize the risk of different therapeutics, but also how do we create an incentive where things that could have a massively positive impact are incentivized to be pushed forward by the FDA?
Yeah. No, I think because there's this common idea of like, oh, maybe biotech companies want the FDA to like go away or not exist. But maybe somewhat counterintuitively, I think personally, the FDA is critical because if we think about, I don't know, like the supplement industry or something, there's no regulation. So it's really driven by whatever podcast you listen to or, whoever you trust, for better or for worse. But it's a marketing game, right? And I can tell you, scientists are often not the best marketers. So organizations like the FDA are what really enable the amount of capital investment that's needed to actually build an efficacious drug to happen. Because if you just allow anything to kind of come to the market, I mean, you see it, right? Like, oh, this water that got classical music played to it, it's going to cure your cancer. And hey, look, if I have an uncurable cancer, I might believe that too. I get it. But that's going to crowd out the ability for someone to actually spend $100 million, $1 billion to build a drug that actually could cure that cancer. Like, that's the only thing that makes these types of investments make sense, is having these kind of third parties that can arbitrate, okay, this is safe, this is effective. And I think something that's maybe lost with, like all the current cost cutting in the government, like DOGE, et cetera, is, actually the pharma companies help fund the FDA's review of drugs. Like, it's not something that's done for free. It kind of is a third party that pharma companies pay to be an arbiter of what works and what doesn't. And you could argue downsides of that, like, oh, pharma's paying the FDA or whatever. But I think most people argue the other way of actually the FDA is too strict. And I think that's incentives, right? The FDA, you get probably promoted for making sure that a risky drug doesn't make it to market and not the counterfactual of, oh, you didn't approve this drug that could have saved a bunch of people, because you never know.
Yeah.
And I think maybe some economist probably has a solution to this, but how do you incentivize that side? I can give you a bad answer, which would be, hey, give the regulator a cut of the profits. Or something. That's a bad idea, right? Too much incentive to approve things. But I'm sure there's some way to counter the fact that I think it is a very risk-averse framework of, hey, just if you didn't approve anything, then no one's going to die. But if you don't approve anything, no one's going to be saved either. So I think there's a lot of innovation that can be done there for smarter people than me to figure out. But I actually, coming from a biotech company, maybe surprisingly, I find the FDA to be actually a critical organization to allowing companies to exist that are building real things.
It's a pretty rare opinion to hear, and I love the measured thinking on that, where it's like, hey, we do need the safety, we do need an external auditor to sort of put a stamp on certain milestones, and I think that's exceptional. But in a case of a company like yours, you are building platforms that make the targeting of specific genes safer, easier to monitor and log. And I would argue, if we're using Cas9, for example, which you guys have done an incredible job building, perhaps that as a platform makes it such that future therapies are safer and safer. So do you think that companies should go through that entire process, start to finish, again and again and again, if we know that there's a platform that's been built that's been shown to be safe for other targets?
Yeah, well, it's funny because you actually hit on one of the main recent innovations of the FDA that this guy, Peter Marks, did. It's no longer there. Which was there's this idea of, hey, there's all these platform technologies, like CRISPR being one of them, of course, but even, antibodies, gene therapies. And for every drug, you're keeping almost all the components the same, and you're just changing one component. So for CRISPR, it's this thing called the guide RNA. And it says, okay, you're going to go to this location in the genome or some other location, but everything else is the same. So why do you need to redo the full safety and everything if we know scientifically that it's really not going to change anything about it? And we're not saying lower the safety bar. We're just saying don't do duplicative work that is just completely unnecessary. And that's this idea of kind of platform approvals, where building your first drug is the hardest, and then your second one's easier, third one's easier.
I love this idea of, let's not lower the safety bar, but let's set it and then determine
be rational about it,
and build on top of that.
And they can kind of, semi-truck can't really drive to a lot of places, but a smart car can kind of fit in anywhere. And that is really important because one of the main things that's kind of held the CRISPR space back so far, and really been a limitation of Cas9, which is the original one that one of my co-founders, Jennifer Doudna, invented, is that it's tough to deliver it.
Has she had any accomplishments that might be interesting?
Yeah, I mean, winning the Nobel Prize.
Oh,
sure.
Interesting. That's
good
notable.
Very, very much so. And that's an amazing technology that actually has an approved therapy today for sickle cell disease. So CRISPR is a very sci-fi thing, but actually there's already a therapy available in the United States and in other countries that potentially cures sickle cell disease, which is super cool. But that's a therapy that's what's called ex vivo. So that's where you have to take the cells out of the body, do whatever edits, and put it back in. And so that's a very limited application. So you can imagine there's many tissues where you can't do that. It's like your brain, I can't really take it out, edit it, put it back in. It's probably not end well for you. So if we really want to tackle like brain disease, muscle, and other areas, you have to have what's called an in vivo therapy. So this is in the body. So whereas the patient perspective of an ex vivo therapy is they get their kind of cells collected at a doctor's office, and often they have to go through like chemotherapy essentially to make sure the cells, when they're put back in, can do the things they need to do. For in vivo, the experience is very different. You go to the doctor's office, you get a shot, you go home. Over the next few weeks, your DNA is changed by the CRISPR technology, and then you're cured of the disease. And you don't really feel much except for getting better.
It's kind of the holy grail.
And yeah, that's really the promise, is that you can have these one-time curative treatments. And that means that you have to have a way of delivering the technology to the right cells and to do the right thing without doing anything that's not safe. And that's a much harder problem. And one of the keys to unlocking that is having these really, really tiny versions of CRISPR, like Cas9 or NanoCas, which is the only way to actually deliver these with things like AAV, which are the ways that today we can really deliver to these liver, non-blood tissues.
That's actually how I had initially read about Cas9. I was looking into some stuff with AAV9 for, modifying brain tissue for mostly, I'm super ADHD and have been trying to figure out how to create some genetic therapies for that. Mostly been looking at transient protocols using LNPs.
Uh-huh.
But yeah, some of the work that you and the greater team have done has been just really inspiring to see, not just because you're building a platform that other people can use and that you've collaborated with other people on, but also that you're developing your own therapeutics in-house end to end. And so how do you think about the balance there between investing in the platform versus putting time and effort into creating specific therapies?
Yeah, and I think you've hit on actually the fundamental problem of running a biotech company, and really the only thing that separates, I think, the best biotech leaders from the ones that struggle, and that's how do you find that balance between research and development? I think it's a constant kind of dynamic question, and I think the first question you have to ask yourself is, what kind of company am I trying to build? And is it like I'm just trying to build one asset? And I say just not in a pejorative sense, but that can help a lot of people and make people a lot of money as well. But you're kind of building one asset, and then once you hit phase 1 or human data or whatever, you're selling it to a pharma company who's then is going to market it, and you kind of move on to the next thing. And that is actually a lot of biotech, just because most things are not platforms and you can't actually develop multiple assets. So there's no sense in building a company around it. You really should give it to the pharma company because they know how to do that stuff and they do it at scale. But if you really think you have a platform that doesn't just build one drug, but builds, or two, but could build 10 or more, and these are really rare, it's like Regeneron, Vertex, maybe Alnylam is on that journey, Moderna, and these are some of the rare opportunities to build a $100 billion biotech company. And that's very much our ambition at Mammoth with CRISPR, is we see this as a platform. We have these really tiny versions of CRISPR that can actually go anywhere in the body, so we can actually tackle thousands of different genetic diseases and beyond. But if you're trying to do that, you actually do need to invest in research. But the problem is that if you went and just talked to your average investor, they'd say, we only care about the clinical results, especially in a bear market. No one, just don't talk to me about research, don't invest in it. You're judged and you will be valued on your clinical assets. And they're not wrong. It is what it is, because that's kind of where the value of what a pharma company would see in it comes from. So you can say, okay, I've chosen I want to be a kind of long-term great biotech company, platform company. How do I actually invest in platform? So there's a couple ways. One, you can go through a bull cycle, and capital can have low cost and you can just raise a bunch of money. And I think that's a lesson in survival and managing cash, but put that to the side. Okay. You don't control that, but I think the one lesson you can learn is survive and be a good steward of capital. So when the boom times come, you raise a lot. When the bear times come, you are in a spot where you're not desperate. But I think the more interesting answer is that you can look at, unfortunately, the few companies that have actually gone down that journey to being a lasting $100 billion biotech company. And one of the key things they've all done is strategic partnerships. And that works because whereas investors are like, okay, what have you done for me lately on the clinical side? Pharma companies, biotech, they're looking for what's next in their pipeline. And sometimes it's clinical, but they also need to fill their earlier stage pipeline. And they might want access to the cool new platform that you've developed because it allows them to do things they wouldn't be able to do otherwise. So they can be a more strategic investor in the research. And that can help fund your research so that you can satisfy your investors and satisfy your internal need to really make sure you don't get leapfrogged by whatever's next, and that you're actually building 10 drugs and not just one drug. If you really believe in your platform. And the reason why these deals can be done is that if you're truly a single asset company, you can't do these because you're selling the future. You have one asset, and if you partner, right, that's your value, right? So you can't sell the future for just cash today. But if you truly have a platform and you really can believe you can work on hundreds, thousands of things, then it should be a no-brainer of hey, I should work with other companies to build drugs and have a higher probability of that, because the more mature company probably knows something more about drug development than you. But B, you're not sacrificing the future. You've got other things to work on and you can drive those forward. And it's kind of a win-win, positive sum game for everybody. And I think that's one of the few ways.
I like what you're saying on win-win, positive sum games, and being able to work with people who traditionally some may call competitors to actually work towards the same goal and really collaboratively build solutions for patients that are exceptional, I think is something the industry needs more of. The culture that you've created at Mammoth, I think, is quite unique in two ways. One, it's very much a tech company and a bio company at the same time, based in Silicon Valley.
Mhm.
Your background being, I guess, initially you were studying history, but
Yeah.
I know you from your Short Tales
Yeah.
blog where you wrote about nothing related to biology, and it was all statistics,
Yep.
which
Yep.
I found you had these beautiful sort of data science visualizations that you clearly put a lot of work into. The other thing is, it seems like there's this very open, positive sum thinking in the team, and you've attracted exceptional people, one of which is Lucas Harrington. Do you want to talk a little bit more on how you've built culture within the organization and how you get to work with like people of such exceptional caliber?
Yeah, I mean, working with Jennifer and Janice and Lucas, I mean, obviously they're all like incredibly accomplished. And I guess I have a couple core beliefs on that. I think one is that you should always try and work with people smarter than you. And it's old Mark Twain, like, don't want to be a member of any club that'll have me. And like, it's always great to be the smartest person in the room, don't get me wrong. But I think you definitely, if you're kind of growing and moving forward in life, I think you should be feeling dumb sometimes. Not all the time, but sometimes I think you really should feel like you are talking to someone much smarter than you in a certain area. And so I think that's a really important just philosophy in general. And then two is that I think that smart people do gravitate towards not just exciting problems, but important problems. I think often we call things like a bubble or whatever, but I think there's real signaling there of like smart people, I think, have some deep intuition in aggregate of like what are the important things, not just, hot things, but important things to work on. And then three is that I think people do underestimate the power of just reaching out. And I think especially PhDs think like, oh, I did my thesis in this area, so I have to found a company on it. I have a bit of a non-traditional story in the sense of I did my PhD at Stanford in biology, but it wasn't in CRISPR, it was in computational biology. And I think I'm very fortunate that I got to know Jennifer and Janice and Lucas, and it's a very non-traditional story in the sense of like, we met each other towards the end of my PhD before we started the company, got to know each other over a matter of months. I think people have two limiters in their mind. One is like, oh, I have to like do my company on my thesis. And that's fine. Like if you have a thesis that you think is a great company, like obviously Janice and Lucas did, that's great. But that's a very small slice of the world. And I think it could be unlikely even. And then two is, oh, I have to have like known my co-founder for 10 years. I mean, that's fine. That can work great. But I think sometimes you've known someone for so long that you don't have the hard conversations of like, hey, what's my vision for this? What do I want to get out of this? Where are we headed? Whereas if you kind of just get to know someone over months, out of necessity, you kind of have to have a lot of these hard conversations. So I think it's not that it's bad to like do a company on your thesis or to like work with someone you've known for 10 years. It's just, I don't think there should be false limiters to doing something really exciting. And I think there can be benefits to the other side of that.
So you're from Georgia, but you were able to come over to the West Coast and do your PhD at Stanford, meet these incredible people. I know you were basically knocking on a Nobel laureate's door up until you were almost kicked out.
Yeah.
But I'm sure for every, Trevor Martin that's in San Francisco or in Boston, there's at least a dozen others that are currently at Georgia Tech, exceptionally talented people that might be looking at what you've accomplished with your career and hoping to do the same. What advice would you have for them in terms of getting from being, exceptionally talented scientists to becoming founders?
Yeah, no, I think, I mean, maybe the trite answer would be something like, oh, like meritocracy, like the cream rises to the crop, just grind away and like, don't worry, you'll be recognized. I think maybe the most helpful piece of advice though would be, well, first of all, the reality that I think, for example, like I grew up in Georgia. I think Georgia Tech has amazing research that will or could create amazing lasting companies that contribute a lot to the world. I don't think just Silicon Valley or Boston or whatever has some monopoly on talent. I don't know if it's evenly distributed, but it's definitely not peaked only in a couple of cities. And so first of all, there is definitely amazing work, and there's probably a lot of alpha to be had in finding these companies at Georgia Tech, because I think they often are overlooked. But from the founder's perspective, I think the best piece of advice I could give is that maybe actually that networks do matter, and that I'm not going to bullshit you that, oh, just don't worry, work hard, and don't worry, like you'll be recognized at some point. Maybe, but I think actually a very important thing to do, especially if you're coming from a university or an area of the country or whatever that's not as traditionally networked as like dropping out of Stanford or coming out of Harvard or Princeton or whatever. And there, I think actually networking can be very valuable. And I think scientists especially look down on it, like, oh, this is disingenuous or like icky. And sure, especially in Silicon Valley, let's say, people over-index on it. But I'm willing to say this very kind of forward because I think scientists will never over-index on it. And I think often under-index on networks are real signaling. It's not just total fluffery. There's a million things going on in the world, and there's limited, let's say, capital allocators, advisors, just anything in your life. There's limited things, right? So you trust smart people that you're friends with to bring you some sort of filtered view on these are important things. And I think navigating those networks is actually a core skill for someone that wants to start a company. Again, you can overdo it, etc. But I think scientists almost always underdo it.
Yeah.
And I think especially if you're coming from like a Georgia Tech or somewhere that doesn't have currently as strong as the network as like a Stanford or even a Berkeley, I think that can be an incredibly important skill and area to spend some amount of time on, and that can be transformative to you.
I think the power of network effects is absolutely incredible. I mean, part of why I moved from Egypt to the US is to be a part of that, and it's tremendously impacted my life. But speaking of network effects, there's this firm called NFX that has some exceptional investors, including Omri, who has a PhD, James Currier, who writes some amazing pieces of work on network effects, who have invested in your company. What have you learned from them, and how do you think about network effects within the context of building a bio company?
Yeah, no, definitely. We're very fortunate early on to have a lot of great people kind of help us navigate the universe, like NFX and Mayfield and many others. And yeah, in general, I think one of the key learnings is on the company building side, frankly, in terms of just understanding like capital formation, like even hiring people, like networks of advisors. I think these are all things where networks effects just kind of permeate every aspect of your life, basically. But within biology itself, I think there, in what we work on in particular, I think it kind of goes back a little bit to this idea of like platform technologies. And I think in general, biology and CRISPR is a manifestation of this, is moving more towards engineering and less of this kind of bespoke artisanal science that it maybe has historically been. And that means you have the benefits of network effects. But your first drug is the hardest one. Your second one's easier. Your third one's easier. And whether that's the actual technical act of building the drug or that's the regulatory path that you have to go down, I think what's really exciting is that there's even some recent news about an N of one therapy where there was a patient that had a bespoke genetic disease. And in less than a year, I think around 6 months, academics and nonprofits were able to build a CRISPR therapy and dose the patient. And it looks like they have potentially been cured. And that shows that this technology can move so rapidly. And it's really more on the clinical trial side that you obviously have to spend a bunch of money and time, but there's a lot of innovation happening there, whether that's international or maybe even in the US. And I think that's where you start to see the evidence of this, of, hey, we can actually build this therapy very quickly. And we can build it even more quickly the second time, more quickly the third time. And I think we're going to live in a world where actually, and this, I think this is true for technology in general, it's like kind of slow and then all at once. Where I think, 10 years from now, we're just going to be knocking out genetic diseases left and right. And it's actually going to be like, oh, wow, like people had to live with like these really debilitating disorders. Like, why did it take so long? And I think the ultimate example of this is this disease called Huntington's disease, where we've known the genetic basis for like, I don't know, 50 years or something, 40 years. We mapped it before sequencing with like microsatellites. And it's this really debilitating genetic disease where basically with almost 100% penetrance, you die in your 30s. And there's really nothing that can be done. And people have tried to target it with like all sorts of small molecules and things because it's a nasty disease. But really, it seems like you have to target the genetics. And if you can target the genetics, you can definitely cure it. So in my opinion, like genetic medicine and like CRISPR in particular will have arrived when the last patient dies of Huntington's disease, because it's a bit of a black eye on the space that we kind of understand exactly what's going on. But still today, there's not a way of actually curing these patients. So I think that's a great example of like a bellwether for the space.
On the point of understanding what's going on, when you were in the early days of the company, it happened to be during COVID. And while the long-term vision was always to build these really powerful gene therapies to cure things essentially with a single shot. At that time when we were dealing with COVID, there was a real need for diagnostics and detection, and you built Detector during that point of time. Was there anything that you learned through doing that you still hold as knowledge today? It's been useful for developing gene therapies.
Yeah, I mean, I think in general, that was an amazing opportunity to demonstrate the team's ability to very quickly go from research work to something that actually got an emergency use authorization and really kind of show that ability to drive a technology from the bench to the bedside. And I think zooming out though, it's a larger kind of story. I think all startups go through this kind of phase of different levels of maturity in the sense of when you're really early on, any work you do, regardless of field, therapeutics, diagnostics, biomanufacturing, can often overlap and kind of read out on each other because the technology is earlier. So, maybe 80% of everything you do is agnostic to maybe what you work on later. And then as the company matures and the technology becomes more mature as well, that kind of narrows and it becomes more of a one-way door and less of a two-way door. And I think that's part of the natural journey of startups is like early on, you should have a view on what you're working on, but you kind of, it's fine if you pivot. It doesn't matter. Like you think that this is where you're going to end up, but actually it's over here. But don't worry, 80% of the work that you did is applicable to either. But then as the company gets more and more advanced in its stage, these doors become less two-way and more one-way of like, hey, actually, if we're not working on that target, we're working on this target, then actually only 20% of what we were kind of doing in that last mile of development is actually applicable. So I think that's actually just normal part of kind of the startup getting to be more mature is that you have to be a little bit more sure about the indication you're working on. So we were very fortunate that a lot of work we're doing is very foundational and also just, moving forward in general, these like novel CRISPR technologies. But I think that's just part of the natural startup journey is that early on, actually, I think you have a lot of flexibility to kind of play in a lot of different areas, actually, because a lot of the things are pretty applicable across because you're a little bit earlier in the research process.
One of the things that I know you're pretty obsessed with is this idea of modularity. How do you think of that within the organization, having so many different, you have a liver disease project that you're specifically working on. The team is, relatively small, but incredibly a high density of talent. How do you ensure that despite having all these little modules within the organization, that there's still like a cohesive kind of company vision?
Yeah, no, I think that's the challenge of all platform companies. It's like a fractal. No matter how far you zoom out or zoom in, there's all these different projects and choices you could make. Probably the hardest thing is, of course, saying no to something. But I think, yeah, this has definitely been a learning over time. But I think, especially in a platform company where there are lots of strategic choices and like things get prioritized or deprioritized or focused on or advanced or kind of killed. I think one thing that, especially if you have a really smart and capable team, which hopefully all startups aspire to have, transparency around strategy is actually incredibly useful and trust building. So whether you agree or disagree with the strategy, like talking more openly about, hey, this is why this is a priority. This is an area we're not sure about, but we're investing in it for these reasons. And these are the reasons why we think it'll work. It's a bit of an asymmetric bet. And then these are the things that we're actually going to deprioritize now. And here's the reasons why, like this shifted in the market or whatever. And I think early on, especially, we were worried about kind of that type of transparency just because we're like, oh, well, what if it changes later? Or, what if people disagree with it or don't understand? But I think actually with the right team that is very high performing and, very intelligent and understands that, hey, strategy is dynamic. And I'm more trying to trust that management is prioritizing things under some rational philosophy and has a plan under any environment. I think that actually is a very powerful way of, especially in a platform company, dealing with the fact that things can be dynamic. And hey, COVID's a great example. COVID happened, that changes the entire dynamic. Diagnostics is different before, during, and after. Even within therapeutics, like different diseases, like, hey, this is a disease that now has an understood genetic basis, and maybe it didn't before. Or, this is an area where regulation has shifted, either positively or negatively. So I think the only wrong answer here is to have a static strategy, I think. And obviously you don't want to just, be a wind vane and change wherever the wind's blowing. But I think one lesson you can also learn from looking at the history of the most successful companies is that they were willing to adapt. And survival is adaptation, I think. You don't need to be an evolutionary biologist to know this. And of course, extremes can be bad, but I think in general, really embracing this idea of like, hey, we're a bunch of smart, intelligent people. We have a North Star, curing all genetic disease and really being able to edit any cell in the genome at any time and any place. And, within that framework, what are the most important things we should be doing right now?
So I love this idea of strategy being dynamic and at the same time not whiplashing the team. I also think strategy has to change as the company scales. I mean, you've raised hundreds of millions of dollars, done incredible work. I think hundreds of millions of dollars in grants alone, on top of
Strategic and
equity investments from phenomenal investors. Talk me through the very early days and what the strategy's like when you don't have hundreds of millions. You just left Stanford. You're trying to build what is, I would argue, probably the most ambitious possible startup vision from the ground up. How do you deal with such a mammoth problem day one?
Yeah, I mean, obviously, I think capital constraints can be good because it makes you think really hard about what is important. But there's a tipping point where it becomes really bad and you just don't get anything done. So I think early on, that is actually a positive thing. And I think, yeah, the main thing in the early days is definitely, the founding team and making sure that you have a group that really shares the same vision of where you want to head, like what you want to do. And then finding the right investors and advisors and supporters that actually also share that vision. And biotech has to be a pretty long-term vision. I mean, you're going to be basically a giant checkbook you have cash coming in, but it's not recurring usually. And you're kind of money losing, let's say, for a while. But the promise of at the end of the rainbow is that A, you're going to cure a bunch of people of disease, and B, shit ton of money to be made helping people in that way. So I think that finding the right kind of advisor group and investor group that is in it for that long haul, if you're trying to actually build the next great $100 billion biotech company, is really, really important. And just making sure you're all aligned kind of on that long-term vision. And then that's on the macro. And then on the micro, okay, what's the most important thing we can do on the next step of that journey with limited capital available? You have to have both thoughts in your head at the same time.
There's this quote, I think it's something along the lines like, the measure of first-rate intelligence is the ability to hold two opposing ideas in your mind at the same time without, I don't know exactly that, but something exactly. And one of the things you're constantly talking about is like the long-term vision and the short-term vision at the same time in a way that feels very cohesive. I just thought that was super cool. Yeah. Medium term, what do you think are some of the platform shifts that are starting to happen, perhaps on a regulatory side, perhaps on a technological side, that we should be excited about?
Yeah, I think the main one is that in general, biology is moving from this like squishy black box of like scientific risk to more true kind of engineering. And I think there's an interesting point here in terms of like, what's the risk in a startup? So when someone's like, oh, I'm going to build the Uber app, no one's like, but can you build an iPhone app that calls cars? No, everyone's like, yeah, of course you can build it. The question is, does anyone want to use that? And will they pay for it? Whereas in biotech, it's been the opposite problem of, yeah, of course, if you cure a disease, and we can debate, payment structures in the US and internationally, but generally, if you cure a disease, there's some sort of product market fit of people will want it and there's some form of paying for it. But the question is, can you do it? And it's unknown. It's like a research problem, could take infinite time. And I think the really exciting thing is, and CRISPR is obviously at the forefront of this, but it's a more general trend as well, is moving to biology being more engineering and saying, hey, yeah, there's product market fit. If you cure a disease, you can actually build a business around it. And B, actually, this is a bit of an engineering problem. Like, it's not a question of, oh, if I had an amazingly smart team and capital and time, could I build a CRISPR therapy for this disease? I think the exciting answer is yes, I definitely can. And that's not an answer we should take for granted or has been true necessarily historically. So I think that's an exciting trend generally, where maybe you can have the best of both worlds long term.
Genetic disease is definitely a general area that I hold pretty close in terms of being important. My grandma, who was the person I was closest to growing up, had a very infamous breast cancer gene that correct, yes. And I think there could have been a world, perhaps if I had been born, she had been born, sorry, a few decades from now, that could have been edited out. How close do you think we are to targeting some of these hereditary, like, cancerous genes?
Well, cancer is an interesting one because cancer is inherently a genetic disease because it's the genetics of the cell going abnormal. So it's very much addressable by genetic medicine. I think the biggest challenge there, or one of the biggest challenges, has been actually delivering to the tumors and like really getting the technology into the cells that need repair the most. And that's something where I think, for example, having ultra complex systems like Cas9 or NanoCast, where you can use lots of different delivery vehicles, can be very helpful. But I think the good news is that it's something that very clearly should be addressable by technologies like CRISPR. I think it's not the bad news, but the kind of thing to be worked on is that there's a lot of detail in how you actually do that, especially with cancer, where you can have these like tumor microenvironments that make it hard to actually even get into the cells that are cancerous. But again, the exciting thing is like these, you can start to lay out more engineering problems, and hard or not hard, it's not just like, oh, is there a pathway that does this? And like, let's start a 10-year research program to even understand what we don't know, right? It's more, okay, I know what edit I need to do. I know what cell I need to go into. Okay, how do I solve these problems and build something, right? That's the question of money, time, and talent. That's fine. Silicon Valley is used to investing money, time, and talent in important problems. So I think that's the exciting directionality, but it's still, there's still barriers to be overcome.
I think we are going to enter a world where product market fit is no longer what we're going for. It's, we know the problem. We just need to build the thing. And that I think is exceptionally exciting. Shifting away from disease and making sure that we're not suffering, where do you think there's opportunity with genetic therapies for them to not be therapies, but potentially augmentations? And how do you think about the very complicated ethics of that?
Yeah, I think personally, I'm super excited about the potential of this. And this is wholly separate from like, do your kids inherit it? I think that's like a whole different can of worms. But like, let's just assume an adult individual is making this choice. I think the exciting thing is that there's actually a lot of genetic targets where we can actually potentially, whether you think about it as like health defense or enhancement or whatever, but like reduce risk of cancer, or only sleep 3 hours a night, or not get all your hair back and not have hair loss, or whatever it is. So I think that the exciting news is that there's actually kind of a lot of exciting targets you can think about for this type of thing. And I'm personally very excited about really delivering on the promise of a lot of this, with really big barrier being not like, oh, what's the genetic target? Or like even a lot of the like, do people want it? Whatever. Like some people want it, some people won't. We're adults, we can make choices. That's kind of my view on it. I think the most interesting thing though, for companies like Mammoth, is that we are really pioneering the ability to deliver genetic therapies at scale safely. Because when you're dealing with like a really rare genetic disease, the safety bar is very high, but I can tell you for like, I don't know, building muscle or something, it's going to be even higher, because you'd be dosing healthy individuals for something that is not life-threatening. So your safety needs to be impeccable. And not only does safety need to be impeccable, but you need to have shown that the safety is impeccable for it to be, let's say, ethical and right. And I think what's exciting is that by building on the foundation that we have, companies like Mammoth, but building on the work that Mammoth is doing, you can actually see the foundation for actually tackling this stuff. Because I think it could be like, oh, let's go straight into like, muscle enhancement or whatever. Fine. Okay. But I can tell you from a practical standpoint, not even just regulatory, but from a person choosing to take this or not, I think the median person will be, okay, how safe is this? Has it been dosed? Like just not taking for the first time. And I think that means that by starting in rare disease and by starting with these diseases where the safety bar is still incredibly high, but there's a huge unmet need where these people need to be dosed with a genetic medicine, you can build up that safety portfolio and really show, hey, this is not just safe enough for someone that has a life-threatening genetic disease. This is safe enough for anyone who wants to actually get this therapy. And I think that creates a potential huge jumping off point for a company like Mammoth to actually become a leader in not just rare disease or the, life-threatening disease. And there's thousands of things to be done there, but really thinking deeply about like these like more defensive things or cancer, these other areas. And I think that's super, super exciting to me.
I like the framework of like defensive versus offensive sort of genetic therapies. I think if there was like one technology or area to target to create the largest impact on disease and health in general, it would be genetics, But the one area in which it's been seen to be limited is genes primarily impact the soft and squishy stuff. Do you think there's a world where potentially, maybe through Yamanaka factors or combinations with stem cell research, that we can actually change some of the more, like, I guess, ossified parts of our body, or that, like, I guess you understand the gist of my question.
Sure. Yeah. I mean, I think in general, some things are easier than others to like reverse or like, yeah, what are one-way doors and two-way doors in biology, maybe. And there's obviously a lot of exciting work going on in like longevity of like, yeah, whether through Yamanaka factors or some other things, trying to make cells younger, but not too young. So in definitely CRISPR can play a huge role in that in terms of reprogramming cells. I mean, reprogramming cells can be turning genes on and off. CRISPR is incredibly good at turning genes on and off. So I think it can definitely be a very powerful way of addressing longevity, general life as one. But two, I think even for things that you might be surprised by, I don't know, like bone density or things like that, biology is a very powerful thing. And I think that genetics can play a role, whether it's like cognitive health or, yeah, ossification of like bone and things. I think actually, yeah, you'd be surprised at how malleable the human body is, even from a genetics standpoint.
So, in many ways, I don't think there are holy grails to things, but CRISPR has felt pretty darn close. The way in which it's not the holy grail is that it has limitations currently. Some of those you've solved with the different Cas enzymes that you've created. What do you think is the next big thing to sort of crack to unlock a lot more that we can do with CRISPR?
Yeah, I would say it's less technology and more on the clinical trial side. Because when we think about like what's the time and effort and capital that goes into a biotech, like 90% of it, probably more, is on the clinical trial. Like the research, yeah, it's expensive, it's like tens of millions of dollars, but really the clinical trial could be $1 billion. And I think that's where, if there's more innovation, there's thousands of genetic diseases, and I don't think people want to wait around like 5 years for every single one of those. And the good news about genetic medicine clinical trials, unlike a lot of things like, say, obesity or something, where you're looking at like the slope of, okay, this group lost 10 pounds, this group lost 5 pounds, is that actually there, as a former statistician, I love that they don't need statistics. You dose a handful of patients, they're either cured or not cured, and it's actually quite obvious. And I think that's something where maybe there can be more creativity or urgency or whatever it is. And I think there's a lot of work being done internationally. In fact, most of the original CRISPR clinical trials were international, like New Zealand, Australia, things like that. And then they came to the US. But I think the US has a huge opportunity to be a leader in really kind of pushing these things forward. And again, it's not lowering the safety bar, it's actually keeping the safety bar as higher, but really kind of thinking about ways to decrease the cost and increase the cadence, maybe starting with rare disease for things where there really are clear readouts and there really is very clear benefit or not to patients. And for things where there is a clear benefit, really running fast, and things where they're not, killing them fast. Both of those are incredibly beneficial to society.
I like that you talked about your statistics background. Stanford is notorious, both in good and bad ways, for having a ton of computer science students who see themselves going into the startup pipeline. I think there's more people at Stanford building startups than not building startups. What advice would you have to a fresh computer science graduate who wants to make an impact on biotech and views biology as the next big thing to crack?
Yeah, I mean, I think the good news is I think it has like a natural impact factor to it that maybe people don't find from optimizing ad clicks or, things like that. Unfortunately, it does pay less than the ad clicks. There's more money in there.
You don't think we need to optimize our ad algorithms more?
I mean, there's probably, I don't think society's benefit is zero, or actually
just
it
1%,
might be negative.
Just 0.5% more.
Yeah, it might be negative actually in some ways, but it's definitely not largely positive in my opinion. So I think there's definitely like an easy answer in terms of, help the world. I think maybe the more interesting answer would be, I think the problems in biology are trickier and more interesting than the ones in ad optimization or other areas, because I think biology is actually more complex and less understood than most other fields. You can apply statistics or LMs or whatever flavor of the month you want to put on it, AI. And there, I think that can be just really exciting. And I think it means that there's also opportunity for real impact, like from an individual. And I think like a manifestation of that would be, for example, with LLMs, like when you're dealing with like just English or not English, maybe with 26 letters, you might be like, oh, that must be more complex than biology because there's only 4 letters, like A, T, C, and G. But I think an example of how biology is incredibly more complex than your kind of classic language LLM is that I think your context windows are wildly different. If you kind of know the 5 words ahead of a word and 5 words after, you can probably predict that word with pretty good accuracy. But if you have a context window that's small in biology, well, there's like 3D structure of the genome, and actually something at one end of the chromosome is interacting with something at the other. Whereas, like, you don't need to look at chapter 1 to predict the 5th word in chapter 10. It's just not necessary. You can probably just look 5 words ahead. And there's also all these unmeasured things that are sitting on top of the language, like methylation or like all these other kind of unmeasured factors. So I think that it's actually an opportunity to develop even brand new statistical methods that really could drive things forward. So that would be my pitch on, why to not get paid $2 million at a foundation model company and come work for a biotech company. Hey, look, I think in the end it pays off more financially as well, but definitely a salary cut. But I think it's just a lot more interesting, both from an impact perspective and from honestly the problems you're working on.
I don't think this salary cut thing will be a thing for long. Once the research cycles are compressed, once we fine tune some of the regulatory frameworks, once we have, you're talking about virtual, I keep blanking on the
Virtual
clinical trials. Once that stuff is, more and more optimized, there is a world where building a biotech company looks increasingly like a software company, including the speed.
Yeah, I think that's that shift towards engineering. And we're not like at that end state yet, but we're definitely on the journey. And that's the exciting time, right? Like once we're at the end state, whatever, and knows it. And then once we're too far from it, there's no point because it's like too far out. But I think the exciting thing is we're in that transition right now. So there's a lot of hay to be made in terms of like being a part of that transition and helping actually create it.
How can startups and companies in general work with Mammoth, and where do you think are some of the best places to get started?
Yeah, I mean, we have obviously jobs posted on our website, but aside from that, my email is public, just trevor@mammoth.bio. And I definitely always love like getting emails from people either thinking about working at startups or starting a startup or going to industry or academia or whatever. And I definitely benefited a lot personally from people just answering random emails from an unknown student. So I try and pay that back to the extent I can. And I think that can be incredibly valuable just in general. And I think one of the great things about Silicon Valley that was surprising to me is that there actually is a lot of people that play this kind of positive sum game of, hey, like, yeah, I don't have equity in your company. I don't even know who you are. But especially in biology, I think people just want to see really cool shit actually come into existence and help people and like cure people and diagnose people. And I think that that's something where, especially coming out of academia, where it's this like pure ivory tower that's not in that messy business world and, so much more enlightened than I think one of the shocking things is that actually I think academia can be way more cutthroat than business, because in academia it's a zero sum game. Like there's a first author and then there's everyone else, and credit is like a bit of a zero sum game of like who's getting credit or not. Whereas in business, like, okay, yeah, you can look down on financial motivation and stuff, and that's not the only motivation, but it is helpful that like everyone can be very successful, and it's not just a total credit game of like, oh, am I first author or last or whatever. So I think that actually people in business can be, in my opinion, more collaborative than people in academia. And I think people are more willing to just reach out and help out and just answer questions without any expectation of return. I try and pay that forward as well.
I know you were knocking on a Nobel laureate's door who is now your co-founder up until you were basically told to stop. What motivated you to keep doing that and have that sheer persistence? And is there any personal reason why you decided that the mission that you're pursuing at Mammoth is the one you want to dedicate your life and a lot of other people's lives to?
Yeah, I mean, I think in my life in general, I try and think about what's the highest impact thing I can uniquely help with. And there's some things that can be helpful with, some that I can't. But definitely this intersection of business and technology and biotech and genetic medicine is definitely something that felt like an area that I can actually contribute. And then if I make that choice, then I want to work with the best people. And that, because I think another lesson I've learned is everything's hard. Like whether you're starting a lemonade stand or you're trying to start like a unicorn company or whatever, it's all hard. And maybe some slightly harder than the other. Maybe the lemonade stand's harder. I don't know. But you really should swing for the fences and really try and work with the best people you can, work with the best technologies you can, and like just not compromise in any regard. Because I think that you are going to dedicate hopefully like 10, 20 years of your life. You're going to spend, nights and weekends, and really, if you're doing the right things, go all in on it. And I don't think you want to go all in on something where it's like you're like starting out from second place and like, I hope we're good enough. Whether you succeed or fail, I think you really should have that ambition of like, okay, this is something that's important. And, whether we succeed or fail, we're going to shoot for being number one. I think that's an important mentality.
I love this polymathic view towards building companies and thinking about in what ways can I create something that others can't, not just by being an expert at one thing, but the intersectionality of things that you're interested in, I think is very unique, from history to statistics to biology. Just combining those things together, I think is super rare. And I do hope that more and more PhDs look at company building as a valid pathway. What do you think is the most powerful part of academia that the startup world can learn from?
I think the biggest trait that PhDs that have done like good PhDs really have as an edge in the startup world is that they're used to things not working, hearing no, having to like just grind it out. I think there's an incredible resilience that you'll find in someone that's done a PhD, where for like 5-6+ years, they just did massive amounts of work that went nowhere. And then it was just the one yes or the one thing that worked that then made all of that worth it. And I think startups, like obviously, hopefully you don't get 1,000 no's for one yes, but you often do. And it's definitely something where I think that grit and resilience is probably the number one thing that a PhD has an edge on. I think on like your average, I don't know, college dropout from Stanford or something.
Yeah, I mean, it's a lot of work for not a lot of pay for a long time.
And you also not paid anything, so you don't mind taking like low salary, and like your opportunity cost is very low because postdocs don't pay much either. So
I guess on a parting note, what was that first yes that felt most meaningful to you?
Oh, definitely my co-founders. Well, and like getting support from like NFX and Mayfield is definitely, the thing that catalyzed it like into like a real company. But I think getting that alignment with the co-founding team is like, definitely the seed that creates.
It's an epic co-founding team, and congrats on everything you've done. I'm super excited to watch Mammoth keep growing, and it's incredible that you guys are tackling such a hard problem.
Might as well work on hard things. So.
Hope to see you soon.
All right. Thank you for having me.
Cheers. Such a pleasure.